This is the third video in the cellular energy unit. In this video, we'll be discussing the role of enzymes in catalysis of cellular chemical reactions. In previous videos, you have learned how to tell if a reaction is spontaneous or not, depending on the free energy of the reaction. However, just because a reaction is spontaneous, that does not tell you anything about how fast the chemical reaction will occur. Left on their own, many chemical reactions necessary for life would take hundreds to thousands of years to occur. They must be sped up in order to occur at a rate that will support life. The function of enzymes in a cell is to speed up a chemical reaction. If we go back and look at our graph that we used to explain exergonic reactions, we will find that it was not drawn completely accurately the first time. In fact, even spontaneous exergonic chemical reactions need a small amount of energy put into the system before the reaction will occur, making the graph look more like this. This energy put into the system is called the activation energy. Also designated EA. The activation energy is used to generate an unstable transition state of the reaction. The lower the activation energy of a reaction, the faster it will occur, because the less energy is required to get the unstable transition state to form. Enzymes function as biological catalysts by lowering the activation energy required by a chemical reaction. In this figure, you can see the difference in the EA when an enzyme, the red line, is added to the chemical reaction. Enzymes reduce the activation energy by properly positioning the reactants and stabilizing the transition state. Notice that while the enzyme speeds up the chemical reaction, it does not change the reaction itself, as the reactants and products are the same. This means that the change in free energy remains constant. When an enzyme interacts with the substrate, it does so at a location called the active site, using an induced fit model of binding. The active site is similar, but not identical, to the size and shape of the substrates that it will bind. Once the substrate binds, the active site changes shape slightly to accommodate the substrate. The substrate is then converted into product while still bound to the enzyme. In this example, the bond between the two monosaccharides of sucrose is broken, separating glucose and fructose. And then the products dissociate from the active site. Notice that the active site goes back to its original conformation after the product is released. The enzyme is not considered a substrate or a product because it is not changed by the reaction. Enzyme active sites form based on the 3D structure of a protein. The tertiary folding of protein structure brings specific amino acids together to form the active site of the enzyme. The active site amino acids may be spaced far apart on the primary sequence of the enzyme, as seen by the purple shaded amino acids in this primary structure. But when the protein is folded, those distant amino acids come together to form the active site. The interactions between the amino acids in the active site and the substrate are weak non-covalent interactions, or sometimes temporary covalent bonds which stabilize the transition state, decreasing the activation energy required. These interactions are highly specific, so enzymes themselves are highly specific for both the substrate and the reaction that they catalyze. Enzyme activity, or the reaction rate, is defined as the amount of substrate that can be converted to product in a given amount of time. As you can see in this graph, as the amount of substrate increases, the rate of the reaction increases for a time. At low concentrations of substrate, there is a lower likelihood that the substrate will bump into the active site of the enzyme. For example, if you have 4 molecules of enzyme and only 1 molecule of substrate, no reaction can occur until the substrate randomly bumps into the enzyme's active site. However, if you have 4 molecules of enzyme and many molecules of substrate, there is a higher likelihood that the substrate will interact with the active site and the reaction will occur. As you increase your substrate concentration, you get to a saturation point where all the active sites of the enzymes are being used, so adding more substrate no longer speeds up the chemical reaction. The reaction rate of an enzyme can be influenced by other molecules which interact with the enzyme itself. Inhibitors are molecules that can decrease enzyme activity. There are 2 types of enzyme inhibition, competitive and non-competitive. In competitive inhibition, a portion of the inhibitor has the same size and shape as the substrate. Notice these 2 regions between the substrate and the enzyme inhibitor. This means that competitive inhibitors are able to form similar interactions with the active site, so they bind to the enzyme's active site instead of the substrate. When this occurs, the substrate is no longer able to bind, and no chemical reaction to produce product takes place. Although the inhibitor binds, it is not catalyzed by the enzyme. This is called competitive enzyme inhibition because the substrate and the inhibitor are both competing for the enzyme active site. Non-competitive inhibitors also slow down enzyme activity, but they do so by binding to the enzyme at a site different than the active site. Binding sites on the enzyme that are different from the active site are called allosteric sites. Non-competitive inhibitors bind to the allosteric site, resulting in a change in the tertiary structure of the enzyme. Notice the change in the shape of the enzyme with and without the inhibitor bound. Oftentimes, the substrate can still bind to the active site, but the change in shape now prevents the enzyme from catalyzing the chemical reaction, so no products are formed. Enzymes that are regulated with non-competitive inhibitors are called allosteric enzymes. Non-competitive inhibitors are most commonly used inside the cell in a process called negative feedback inhibition. Feedback inhibition is important for enzymatic pathways to regulate the production of the pathway's end product. Look at this pathway where molecule A is converted to molecule B by enzyme 1. B is then converted to molecule C by enzyme 2, and C is converted to D by enzyme 3. When the cell has produced enough of molecule D, it does not want to continue wasting energy and resources to make more. Molecule D can act as a non-competitive inhibitor for enzyme 1. This shuts off the enzyme in the first step of this pathway, which will stop the production of the remaining pathway products. If we look at the shape of the products, this makes sense because the end product will often not look like the starting product, so competitive inhibition would not work. Often, metabolic pathways in cells are highly branched. When this occurs, the end products of the reactions inhibiting the first enzyme in the pathway that is unique to them. Enzymes can also be activated by interactions with other molecules. In this case, an activator protein binds to the enzyme's allosteric site. This again changes its conformation, but in a positive way. The new tertiary shape of the enzyme is better able to bind the substrate, increasing the rate of the chemical reaction.